Okay, I think we'll get started. Welcome everybody to our special seminar today. It's a pleasure to have Dr. Luis Fernando Queme Cobar here. Dr. Queme is a research associate at Cincinnati Children's Hospital Medical Center, where he works in the Department of Anesthesia. Prior to that, he did his undergraduate medical degree, initially in Guatemala City at Francisco Marroquin University, and then did his doctoral work in Japan at Nagoya University. And from there, he moved to the States, to Cincinnati, where he originally worked as a postdoctoral fellow in the same place he's at now, and then went on to be a research associate there. As a postdoc, he worked primarily on modulation and nociceptors and cardiovascular reflexes by muscle afferents, particularly in response to ischemia. And then most recently, as a research associate, he's developed a chronic muscle pain model for repeated injury, looking at things like changes in gene expression in the peripheral nervous system as well. And I should add that he's here today as part of a program at Virginia Tech known as the Future Faculty Development Program for scholars that are traditionally underrepresented in their fields. And he's a guest of both the medical school, the Virginia Tech Carilion School of Medicine, and the Fralin Biomedical Research Institute here in Roanoke today. So we're very excited to hear about his work, and he's going to tell us about molecular mechanisms of peripheral sensitization and ischemic myalgia. Welcome. It's good to have you here.
Thank you very much for the kind introduction. Today, what I want to talk about and try to show you it's how primary sensory neurons show gene expression plasticity, and after a very specific type of injury, they change their gene expression in response to signals coming from outside the neuron and can influence how pain is perceived. So to start a little bit with where we're going, I would like to talk a little bit about pain. And when we define pain, I like to follow the definition from the International Association for the Study of Pain, which is that it's an unpleasant sensory and emotional experience associated with actual or potential tissue damage or described in the context of such damage. And they're revising this definition because pain, it's something that is not just sensory. It involves a heavy emotional component, and in order to better understand how it's working, we need to understand that it's happening at different levels in the system. I focus on the primary receptors, but it's important to know that our models have definite limitations when we're looking at this. When we talk about chronic pain, we're definitely talking about pain that has lasted more than 6 months. And it is one of the most common reasons why adults seek medical care. It's linked to a bunch of different comorbidities, restrictions in mobility, obviously opioid addiction due to the treatment choices we have right now to do that, anxiety, depression, and in general, poor predictors of health for patients. And chronic pain has always been thought more of like a side symptom of what's going on with the main disease. So we never really, we treat pain as the extra thing that's, it's become the side dish of the disease instead of being something that we should be more focused on. What's the impact of pain in society? Well, in 2016, a survey from the CDC looked at about 1 in 5 US adults experience some form of chronic pain, and about 8% have what they call high-impact chronic pain. It means pain that has enough disability that it will impair your mobility and your everyday life. So it has a huge impact. And when you put cost to it associated with, let's say, loss of productivity, cost of medical care, all of these things, you can get something of up to around $635 billion, which is more than what the cost of heart disease, cancer, and diabetes. So in that sense, pain is a huge problem, and as it can be reflected in the current state of things like the opioid crisis, it's something that it's not being properly addressed in the field. If we focus more in musculoskeletal pain, this is one of the most common types of pain It has different causes. Exercise is what most people are familiar with. You work out, have an intense workout, and now you're feeling pain after it. People as they age, they start feeling more pain. Trauma definitely is a cause. Inflammation, depending on different types of diseases. And in this case, I want to focus in ischemia. Ischemia is an underlying factor in a variety of conditions. Things like sickle cell anemia and peripheral vascular disease will come to mind immediately. It's the traditional things. Tourniquet pain, it's something that also involves in ischemia. It's frequently observed situations like orthopedic surgery, where you actually go and block the blood flow of the whole limb before you can actually do a major surgery. And there are more difficult situations like complex regional pain syndrome or fibromyalgia, where the involvement of ischemia is not very clear, but we have observed that our patients have a very clear vascular component where they have perfusion deficits and anomalies that can be associated specifically with the sore areas in the tissue. So in this sense, studying ischemia as a cause of pain is extremely relevant. And when I study ischemia, I like to focus at the root of the cause. And for me, that is, where does the signal start? And I just go back to the primary sensory neuron. It's your pain nociceptors in the skin. And in the muscle, it's what we would call our group 3, 4 afferents. These are or slightly myelinated or unmyelinated afferents, basically free nerve endings inside your muscle tissue. And these respond to a variety of stimuli. They respond to changes in temperature, heat, cold, mechanical stimulation, basically pressure, and chemical stimulation, which personally it's what interests me the most, because it's a very varied stimulus. Chemosensitive afferents in the muscle do not respond just to one type of thing, like low pH or, let's say, lactic acid, which is what most people hear about with exercise. To actually get a response, you need to have a combination of these factors stimulating the afferent at the same time. And studies from Light in Utah has shown that you activate very specific subpopulations of sensory neurons if you have different concentrations of all of these metabolites. So in that sense, group 3 and 4 muscle afferents are very interesting, but they also have an extra function to complicate the frame a little bit more, and it's that they are the sensory arm of cardiovascular reflexes to muscle activity. So you work out, you get increased heart rate, increased respiratory rate, a little bit of increase of blood pressure normally, and that can also be partially modulated by the sensory signals from your group 3, 4 afferents. Now, I want to put a little bit on the side, the cardiovascular side of the role of primary muscle afferents, and focus on what happens with pain and what happens when we have an ischemic insult in the muscle. So we wanted to study that in animals, and our animal model for doing this involved doing occlusions of a major artery in a limb. So to do that, we do a small surgery in our mice and tie a suture around the brachial artery. And we developed 2 types of injury models because we wanted to see if there were any differences in how afferents would react to either a prolonged injury, which is what we would call the brachial artery occlusion, where we just tie the suture, leave it there, and we look at the animal 24 hours after the injury, or an ischemia with a reperfusion injury. In this case, I'm more interested in that one because I feel it's more relevant clinically. You do not usually see people that completely lose blood flow on a limb and it just stays like that. What you see is small bouts of occlusion, reperfusion, and that's what you're usually observing. To characterize how the injury works, we recruit our friendly mice, and we do a battery of assays. So if you remember when I was talking about pain, you cannot describe pain in an animal very easily. You need to look at it comprehensively. So the way we do that is we look at reflex behaviors that can tell us if you're actually having some sensitization in your afferents. To do that, we use a stimulation with bone-fried filaments, and there's a sneaky rat pretending to be a mouse in this image. But we also look at how the animals behave when you just let them be at rest. And to do that, we use guarding assay where we look at how much the animals bear weight on their injured limb, and we just score it on a scale, try to figure out how it goes. And we also look at the muscle function. So for that, we measure the grip strength and taking a page from muscle biologists and just try to see if there's changes in the muscle function, because increased pain can decrease your motor neuron ability. So we can take that as an image and try to have an idea of what's actually going on in our animals. So when we look at the behavior of our animals after injury, and this is some work that was done very early in the lab, almost right after I joined, we looked at this are 3 behaviors. And I'm going to move around. That's kind of my easiest way to explain. We look at our guarding assay, basically our injured animals, and you can barely see what happens in our sham injured animals. It's basically no change in how much. They are always bearing weight in their 2 paws, or injured animals 1 day after injury, either by or brachial artery occlusion or ischemia with reperfusion, where we just tie and remove the suture, they develop pain. But at 3 days, they seem to be recuperating faster in our ischemia with reperfusion injury. We look at their grip strength, and they show decreased grip strength, obviously, 1 day after the injury. And later on, they do not improve that well. And I know this graph, it's really busy, but if I try to make the point here, it would be just that our animals are also having decreased withdrawal thresholds. So we had to poke them with less strong filaments, and they will withdraw faster in this test. And this is just to make the point that after the injury, the animals get sensitized, but also that different types of injury on a long term have different behavioral characteristics. So, okay, this is being interpreted as increased afferent activity. And to look at how we actually, at what's going on with the afferent, well, my mentor developed this preparation. It's an ex vivo preparation where we basically dissect all the muscles in the forepaw of the mouse, and we keep our nerves connected specifically to the median and ulnar nerves all the way to the dorsal root ganglion and then to the spinal cord, so we can record directly from the neurons and the dorsal root ganglion. If you see that little glass thing that you can see over there, that's our sharp tip electrode just going inside of the DRG, and we can stimulate the receptive field of our neurons in the muscle tissue and just look exactly at the activity of these afferents in the muscle. And obviously, we can apply different types of stimuli. Thermal and mechanical are the easiest to do, but to do chemical stimulation, we also have this tiny little separated chamber around the muscle where we can change all the solutions that are surrounding the muscle tissue without exposing the dorsal root ganglion neuron to them. And we can look at what's going on over there. So to try to show some of our quick finds on this is that when we do heat stimulation of our neurons, there's increased peak firing rates for our brachial artery occlusion model, not as intense in our ischemia with reperfusion. It's just telling you that neurons are firing faster when you stimulate them to heat. So they are more sensitive to heat stimulation. You look at our mechanical threshold, which is, again, analog at what we did with our behavior. Now our neurons are responding when you apply less force to the receptive field in the muscle. And this is probably my favorite graph in that paper from 2014. We looked at their chemosensitive properties. And what we observe in both either injured animals, it's very similar. So I want to show you more or less what's the usual response of chemosensitive neurons in the muscle. We try to categorize the responses in what we call low metabolites. So this is something about pH 7, which is not very acidic, lactic acid in the muscle, combined with a little bit of ATP and about 5 micromolar. It's very little. It's not enough to actually induce any pain in humans. There are studies where they prepare these cocktails in you that they inject them into patients, and what they report is fatigue in their muscles. So we are interpreting this low metabolite concentration as what you would see in fatigue. And you have a very specific subpopulation that responds exclusively to low metabolite concentrations. Now, when we apply the high metabolites, which is now about 50 micromolar ATP, pH all the way down to 6.6, which in the muscle is extremely low. And now you can see that there's, again, a very specific subpopulation that goes just too high. There's almost no neurons that respond to both. So we think that there are sensory-specific modalities between our neurons. But after our injury, what we observe is less neurons that go specifically just to low metabolites, and now there's this huge subpopulation of neurons that are responding to both low and high metabolite concentrations. So this chemosensitive neurons lost their capability to discern between what's supposed to be a noxious and non-noxious stimulus. And that's where it gets more interesting, because it could explain why our animals are just spontaneously, if you want to call it like that, withdrawing pain. They are feeling pain all the time, because now their neurons, instead of sensing fatigue as they should, they look like they are sensing, and they're coding for pain. So what is going on behind that? And this is just to signal that we looked at a lot of genes that are related to pain perception in general. But I wanted to make the point just about what's going on in the muscle and dorsal root ganglia. And in the muscle, we see two really important signaling molecules going up after injury. One, it's interleukin-1 beta, and the other one, it's glial cell-derived neurotrophic factor. So one growth factor, one interleukin, both have been associated to increase pain in different models. But what was more interesting is that the receptors are specifically upregulated in the dorsal root ganglia that were affected, and they were innervating the affected tissue. So now that we know that we had this, we kind of had two really good targets to go after. Try to figure out if the signaling molecules are what's behind this increased afferent sensitivity. Now, part of my work during my PhD was looking at pain after exercise. And when we were doing that, we also found that GDNF was an important signal to developing the delay onset muscle soreness, which is just a pain that you get after working out if you're not used to it. And we thought, okay, if GDNF, it's also in this, and then now that we saw this specific signature of GDNF signaling in this other model, I told Mike, you know what, I want to go after this. So can I look a little bit deeper into what GDNF is doing in our model? And since I'm more interested in the ischemia with reperfusion model, and we see a specific gene upregulation in this model, let's chase after it. And the first thing I had to do is to show that we actually had GDNF in the muscle. So did some Western blot experiments, just quantifying the amount of GDNF after injury. And what we can see is that our injured animals indeed have increased GDNF in their muscle. And then we did immunohistochemistry, and this was a strike in the moment that I put them in the microscope. So I just got the slides, and I put one of the slides under it, and it's like black, and I thought, wow, it didn't work. The experiment is a bust. This antibody is terrible, and we cannot actually stain for GDNF. And they hand me the next one. It's an injured animal, and now you see this beautiful kind of red halo-like structures around the myofiber. I was like, okay, definitely there's something in there. So we are sure that GDNF it's been increasingly expressed in the muscle tissue, and now we're starting to believe that it may be coming from the myofiber. We're not completely sure about that, but so far our data is pointing us in that direction. We look at the dorsal root ganglia and the expression of GFR-alpha-1 just to verify what we did in our previous experiments, and indeed GFR-alpha-1 is up in the DRG. So now I want to target the increased expression of the receptor for GDNF, because I think it's a really good way of showing how upregulation of a single receptor in a cell can change all the gene expression downstream. GFR-alpha-1 being a growth factor pairs with red, and it has a very a wide signaling cascade under it. It can definitely modulate gene expression downstream of it. So how do we do that? When Mike was doing his work in Pittsburgh, he developed this really clever strategy to do RNA interference in only the cells that are innervating the affected tissue in the injuries. And what he did is he linked this transfection factor, it's called penetrating 1, to the siRNA specifically targeting or the gene that we want to express. And it just gets, we can that way injected directly into the nerves that innervate our affected tissue, let it transport retrogradely, and we do this 2 days before doing our injury protocol just to give it time to get back to the dorsal root ganglia. Theory, it should prevent the upregulation that we see after injury. So just by doing that, we corroborate that our knockdown works, and indeed I was very excited. It was one of the siRNAs that we tested was very effective at bringing down the levels of GFR-alpha-1 receptor in the dorsal root ganglia back to normal levels. So, okay, how is this affecting behavior? And we repeat all our battery of tests that we did before. So we look at the guarding scores. And one interesting thing is like, if you look at our red arrows, and everything that is red here is going to be our injured animals, or yellow, it's a non-targeting siRNA control, just to make sure that our injection is not what's modifying things, and it's not just putting random siRNA inside of the cells, what's modifying them here. And what we can see is that it can dampen the increase guarding behaviors. It changes, prevents the development of mechanical hypersensitivity in when we stimulate with bone fry. I was not very satisfied because bone fry is very superficial. It's skin. We're looking at muscle. So we also did a muscle squeezing test where we have this blunt probe just to try to squeeze the paw of our mice. And we can also prevent the increased mechanical hypersensitivity to muscle squeezing. And now I'm a little bit happier. It's going really well, but we cannot completely recover the increased muscle weakness after injury. So, okay, that's really nice. But it makes sense that if we are affecting all the sensory capabilities of the neurons and they are hypersensitive, and these neurons have two different functions, and this cardiovascular reflex sensory arm function, it's modulated by their chemosensitive capabilities, we should be able, there should be changes in this. And this, yeah. Sure. Yes.
So I want to make sure I have the construct correct. So you get increased firing rate of the sensory neuron in response to the stimulation, the nociceptive stimulation. Now that increased firing rate may be contributing to changes in expression, whether it's immediate early genes or GDNF, whatever, in the ganglion cell itself, correct?
So
Or not?
basically we think that the increased firing rate of the neuron, it's the consequence.
That's what I was trying to get at. So your hypothesis is that the change in gene expression is then causing secretion binding to a receptor, and that in some way is modifying thresholds, ion channel sensitivity, trip channels, whatever it might be, to cause it to fire more.
That's exactly where we're going at. So basically, it's signal coming from the muscle, and the injury per se, it's inducing changes in gene expression in the neuron. So now the neurons, as a consequence of that, firing faster, and we're trying to tease out why is that this is going on.
So as a control experiment, have you done anything to drive the firing of the sensory neurons independent of this, just as a baseline to show if you drive a DRG cell to fire at 100 hertz versus 10 hertz resting rate, does that change anything in terms of expression, independent of this, just firing per se?
Just make the afferent fire per se. No, we have never done that type of study. We think that in this case, the gene expression might be kind of one of those self-potentiating signals, specifically because we're looking at GDNF. And red, it's a very quirky receptor by itself. And it's been seen in different models that signal that activates red receptor can potentiate the gene expression of the receptor that's binding red. So we're thinking that that might be what's behind, specifically with GFR-alpha-1. We don't have it that completely teased out yet, so I'll be lying to you if I tell you I'm completely sure about that. So trying to move forward and show what we want to see. We want to see if our hypersensitive afferents are actually affecting the cardiovascular sensitivity. To do that, we had to develop an exercise protocol. What you usually see in most experiments that look at cardiovascular function mediated by sensory neurons, it's you have an anesthetized animal or an surveyed animal, and you just expose the nerve and usually zap them, and that's not very functional in my feeling. I've always tried to do my research trying to look at a more natural stimulus. So what we did is we developed this very mild exercise protocol. We put our animals in like one of these metabolic chambers, kind of an oximax system, to try to measure how much CO2 they are putting out, try to make sure that our exercise protocol is mild enough that you would observe you're exercising the animals, but you're not pushing them to the point where it's ischemic contractions. So what you can relate this as what people that study metabolism call the lactate threshold. Once you break that, you're basically getting ischemic contractions, and you can extrapolate that just from measuring the CO2 that it's being outputted. So we developed this protocol in this chamber, and then we put our animals in a treadmill following the same intensity of the exercise, and we measure their blood pressure before and after exercise. And what we see here is that in our sham-injured animals, we could not see any changes in blood pressure. So that initially made me feel very upset because you have a natural reflex. You should see an increased blood pressure in sham-injured animals. But when we didn't do that, observe that, it's like, I don't know if I should keep pursuing that. Looked at the injured animals, and now our injured animals did show an increase in their mean arterial blood pressure, but only after exercise. So just by doing the injury per se, that's not enough to actually drive their blood pressure up. You had to exercise them while they are injured to actually observe changes in their mean arterial blood pressure. But obviously, someone was going to ask, are you sure that this is not artificial? How do you know it's a real reflex and everything? So we also did the catheterization experiments where we put the animal under anesthesia, and we directly stimulate the afferents with an electrical stimulus just to activate them and make sure that we can see a cardiovascular response. And indeed, we can see in our sham-injured animals a normal cardiovascular response, but our injured and controlled animals have an increased response to the muscle contractions. What we see here is just the increased blood pressure that you observe inside of the 30-second parameter time of stimulation that we give them. So what you would see is just your animals having muscle contraction where they are just kind of clawing their paw as hard as they can for 30 seconds. You let go, and you see changes in the blood pressure. And
when you're stimulating the nerve and you have group 1 and 2A afferents as well, which have lower thresholds, how do you certainly attribute the blood pressure effects to the group 3, 4 higher threshold fibers?
We try to do that. So what we do is, well, to stimulate your motor neurons, you can just stimulate at twitch threshold. And if you do, afferents respond depending on how intense you're giving them your stimulus. So when we try to give the stimulation at just 2 times the twitch threshold, we didn't see any changes in the blood pressure reflex. In some animals, you would see maybe a slight hump, but it was not significant. And we're thinking, okay, this is just what happens if you just have a mild sensory activity or you just have contraction. These are very low. We keep ramping up all the stimulus slowly, going from that very mild stimulus all the way to 1 milliampere. And at 1 milliampere, you know that you need to get at least between 0.8 to 1 milliamperes to engage your group 3 and group 4 afferents. Being slightly myelinated and non-myelinated, you need to give them a little bit more current to stimulate. Yes, you're at that point you're stimulating everything. You cannot tease out, but if you do not get a response at lower intensities, you can assume that evidently this is coming from the ones that were all the way to the top. And we do observe this increase in blood pressure after activating our afferents. So now I'm very excited because I think that I have two different biological phenomena, just pain and increased cardiovascular responses being modulated by one single phenomenon. We did again some electrophysiology to try to better characterize what was going on, and we recorded from our injured animals, and what we see is that the afferents are indeed mechanically hypersensitive, as we observed before, but knocking down the increased expression of GFR-alpha-1 prevents that increased mechanical hypersensitivity. Now, this is kind of a busy image, so I want you to try to focus specifically in purple, red, and light blue, because that's, again, our populations of low, high, and both responders of what we see. And what you can see is that what we have in our naive animals in this case is just a very small population of both responders, almost non-existent, close to 1 or 2% in this case, population of high responders and or low responders. And when we switch to injury or controls, we basically lose a significant proportion of our low responder population, and we have an increase in both responders, again, according to what we have done before. But when we knock down GFR-alpha-1, now we only see this two very distinct subpopulations of chemosensitive afferents. So I'm trying to think that GFR-alpha-1 might be doing something with chemosensitivity. Also something that I do not have in this image, but is that the total number of chemosensitive afferents that I could find in my recordings decreased. And now I'm not seeing a lot of cells that respond to chemical stimulus in my electrophysiological experiments. I'm wondering why is this going on? So, well, our next step was, again, try to just do some PCR after our knockdown. And this is more or less just a combination of or IR and controls, since at this point they have not shown any difference between each other. And again, what we observed is the increased upregulation of GFR-alpha-1. And out of all the genes that we observed, the one that was interesting was P2X5, because when we did the GFR-alpha-1 knockdown, it was the only one that actually stopped being upregulated. So knocking down GFR-alpha-1 can prevent the upregulation of the P2X5 receptor. Now, what the P2X5 is an ATP sensing ion channel, and it has been associated with modulating the sensitivity of acid sensing ion channels to low pH. So basically making an ion channel respond to higher pHs than what it's used to. So now it's kind of all coming together. It is a signal that it's changing how our chemosensitive afferents are responding not just to ATP, but also to a low pH. And that, I think, reflects very well in the phenotypic changes that we observe in our chemosensitive or response to chemical stimulation profiles. Also, we did see a slight decrease in the upregulation of ASIC3, but in this case, I was personally more interested in trying to pursue just P2X5. So I ran some immunohistochemistry experiments with all the DRGs from our preparations, stained for GFR-alpha-1 and P2X5, and what we observe is an increase in the total number of cells that are positive for GFR-alpha-1 in dorsal root ganglia after injury. And when we look at P2X5, it's also increasingly expressed after DRG, and we can bring it back with our targeted knockdown. So, okay, so far so good. The next step is, or was for me, to try to figure out if knocking down P2X5, so far everything boiled down to P2X5 being downstream of GFR-alpha-1, knocking down P2X5 should do exactly the same, or at least partially what knocking down GFR-alpha-1 did. We knocked it down. I was very excited when I saw the image for our paw guarding experiment, because it looks almost exactly the same as our GFR-alpha-1 knockdown experiment. I was like, okay, it's going perfect. And then I started looking at all the other responses. And it kind of just boils down to saying the more downstream you go and the more specific you go, your responses are less broad. Now, we cannot completely revert the mechanical hypersensitivity or revert the decreased muscle strength. But we can very effectively prevent increased mean arterial blood pressure after exercising or injured animals. So this, to me, is also pointing out that this increase in blood pressure that we're observing might be dependent of the chemosensitive properties of these neurons. So the question remains, how do we go all the way from signal receptor all the way in the molecule and the cell membrane of the neuron and back to an increased expression of an ion channel? And this is just to kind of try to show you all the experiments we try to do to pin down the intermediary in the signal pathway. If you ever go to one of those antibody distributor sites that give you all the the signaling charts for all of your things, you'll see that below red, there's a whole screen of things that you can test. And we started trying to go more specifically. At some point, we just decided to microarray, and nothing seemed to significantly change after injury. And at this point, I'm extremely frustrated. We had done some other experiments focusing on IL-1 beta, and we had teased out that junk was the transcription factor behind IL-1 beta. So we're looking for something a little bit different. We did the same experiment with GDNF, and we try to do culture cells, try to quantify them. There's not a significant increase of cells that are positive to phosphorylated junk after exposure to GDNF. So we was kind of puzzled here. I didn't know where to go. And one of the last things that are at the end of the image is this response factor binding protein called CREV. And that's a transcription factor, basically binds your promoter in the DNA, and it facilitates transcription. And it has multiple pieces to actually get activated. To have this transcription factor activated, you need to get CREV bound to the DNA, then you have to have it phosphorylated, and now you have to have a co-activator protein come and bind to it. And I got really excited when I did the first experiments and saw that phosphorylated CREV was actually significantly increased in our injured animals, but we did not, we could not bring it back to naive levels with the GFR-alpha-1 knockdown. So next step, try the binding protein, and it was an extremely challenging experiment being at 250 kilodalton protein. It's a nightmare of a Western blot to try to tease out. Had to change completely all of my research protocols, but when we worked out all the kinks and the methodology, we actually got to observe that CREV binding protein is actually gets upregulated after injury, and it can this upregulation can be prevented by knocking down GFR-alpha-1. So if you need the interaction from CBP to actually activate CREV, blocking this interaction should prevent the or increased pain-related behaviors, and it should prevent the sensitization of our afferents. So a really quick experiment found a specific antagonist that's used in oncology research as was used as an experimental drug to try to tackle leukemia, and we injected it systemically, IP, just try to do our vehicle controls, and what you can see is that by effectively blocking the interaction between CREV and CREV binding protein, we can prevent the development of increased spontaneous pain, and we can partially dampen the increased mechanical hypersensitivity and try to rescue a little bit of the muscle strength in our animals. It was, again, extremely effective at preventing the increased mean arterial blood pressure after injury. And the cherry on the pie is that if this is the mechanism, it should, of course, prevent the upregulation of P2X5, did the Western blots, and it worked out. So it's one of those things that when you, after a lot of struggle, you kind of hit that it works out well moment, and made me feel really excited because we actually had a mechanism that was teased out. But if you remember what I told you in the beginning, I'm all about catching the injury signal all the way in the beginning. If you, it's always been my philosophy that if you want to do an intervention and you block something from the start, you can prevent all the downstream consequences. And I'm familiar with what's being done in other disciplines. We use NGF as an experimental medication for pain in rheumatoid arthritis. NGF antibodies, I'm sorry. And okay, let's try to block this using an anti-GDNF antibody. Let's just inject it locally, low dose, and let's see what happens. So we injured our animals, and at the time of injury, we inject 5 microliters of concentrated anti-GDNF antibody into their injured forepaw. Run behavior can dampen the increased pain-related behaviors, also the increased mechanical hypersensitivity and muscle strength, and prevents the increased blood pressure. So at this point, I feel I have my whole story together. This is just a summary of what's going on. What happens after you get in this very specific branch of signal after ischemia with reperfusion injuries, that you get increased GDNF signal in the muscle tissue. Your injured muscle is releasing, obviously, ATP, lactate, and proteins to have low pH inside of the muscle tissue. And you get, as a consequence, increased pain-related behaviors in our mice and an increased exercise-mediated reflex. This is all happening because in the neuron, what we observe is increased expression of GFR-alpha-1 that modulates the expression of P2X5 by CBP-CREB interaction-dependent mechanism. And that's pretty much where I decided to leave this story, try to get it out. And I would like to focus on what I want to do with it, where I want to take it. So a long time ago, there was this group in Germany, and they put out this paper looking at complex regional pain syndrome. They're looking what could be the underlying cause of this thing. And they're proposing that injured neurons that are more sensitive can modulate the peripheral vascular tone and cause increased peripheral vascular resistance that would cause a vicious cycle of ischemia-like perfusion deficits in the muscle and just enter into a chronic loop of more ischemic injury, more afferent sensitization, more ischemia, and prolong the pain and the people that are experiencing things like CRPS. So, well, we just did a quick experiment trying to repeat the injuries and ischemia with the perfusion. I told you originally these animals recuperate very well, about 5 days. They're doing really fine. It looks pretty much like they're sham injury controls. If you actually do double injuries and you do a first injury, let them recover, injure them again, what you see is that now the mechanical hypersensitivity, and in this case the spontaneous pain that they are showing, does not recuperate as fast. So kind of puts me into this mindset that we should be looking more into this underlying mechanism and tackling the hypersensitivity of this group 3, group 4 muscle afferents could be a viable strategy to prevent the development of chronic pain in some of our patients. Some other future directions of where I would like to take my research of some of the work that we're doing now in Cincinnati Children's. It's kind of like a new project we're starting right now. Is we have one of our colleagues in the anesthesia department is doing this study, and what she observed is that child anxiety, and it's a great predictor of chronic post-surgical pain, and that kids that have more anxiety and report more anxiety in their test also have a higher risk of developing chronic pain later on in their lives. So taking that into context, I'm thinking, how is there an underlying mechanism with this? There have been some studies, mostly one-offs, trying to look at the role of stress into the increased sensitivity of muscle afferents. They show definitely effect of stress, but they have never been able to pin down the mechanism. Some of the studies, they just remove the adrenals, and that's like telling, oh, look, the car is not running, but you remove the gas pedal. Obviously, it's not going to go. So I really trying to tease out the mechanism. I think it might be driven by neuroimmune interactions. So we're developing this stress model in mice where we're using a stimulus that's very typical. We're using loss of enrichment. It's a stimulus where you make the mouse as happy as possible. Imagine someone that has everything, gets a bad luck moment, and now it's, they lose everything. That stresses animals, creates a very stereotypical neuroimmune signature in the brain, just excessive glial activation that shows how it's correlated with their stress. And I think that what we can observe at the primary sensory afferent level is that also this increased inflammatory or inflammation-like state in the body that you get during stress can sensitize your muscle afferents. And if you get this happening over and over again for enough occasions, you know, one thing piles on top of the other, I believe that this could actually tease out better the mechanism of what's going out in chronic pain, taking into account what we observed in populations. And just try to not to run too long. I'm going to skip my last slide and thank everyone from our laboratory in Cincinnati Children's Hospital, just specifically Dr. Mike Jankowski, who kind of took the chance of hiring me right out of the Ph.D. and trying to say, okay, come here, come and play with our mice, learn some of our techniques, and try to move on your research career. Also, our supporting staff, our research assistants that helped me troubleshoot a lot of these experiments, try to get our immunohistochemistry Western blots out. And last but not least, our grad students and magnificent undergrads, because they did a lot of the trial and error work trying to do these things. And without them, I wouldn't have all these things. Also, our collaborators from the molecular cardiovascular departments, because I had no idea how to do any cardiovascular experiments before this thing. And they took time from their days to kind of illuminate me on how to do this thing. The money, of course, because without them, we're nothing. So thank you to NIH and American Heart Association. I'm open for questions. Mm-hmm. Sure.
I have a question. So expert printers in monitoring for ventilation, but I was wondering if you have evidences or if you were thinking of looking at what is happening with patients, if you find the same molecular mechanism, because sometimes when they are not used effectively,
Well, so that's an interesting thing. We have not done the work specifically. The groups that are more involved into looking at what's going on are some groups in Utah that are actually trying to tease out more how neurons respond to this very specific fatigue-like or ischemia-like stimuli. Well, I'm actually looking forward to what they are going to do. So far, they seem to have very nice signatures in their patients. They are doing some really massive sequencing studies using patient samples. And from what I've heard now, what they are seeing is a very, again, specific neuroimmune signature and increased activation of metabolism-related genes in patients. So that.
I was interested in your protocol. Started off using the permanent occlusion.
Yeah.
We'd expect a lot of tissue damage with the necrosis to begin. And your intermittent fusion or ischemic model, how long was that ischemia kept for, and how long was the recovery?
Okay, so I'll start with the long-term occlusion, brachial artery occlusion. So when we tied a suture around the brachial artery, we try to make it as tight as possible, but you are fighting against 120 millimeters of mercury at 500 beats per minute, give or take at any moment. So as strong as your occlusion is, you always get some blood flow going through. So there's reduced blood flow depending on arteries. Different papers report different things. I estimate we would be, give or take, about 15 to 20% of the blood flow that's still going. Also, mice seem to have very nice collateral circulation at the level that we do. So yeah, if you do this experiment, or you would just occlude a major artery in a human, you lose the limb in hours. Mice are way more resilient, which makes them a nice model too. Now, with ischemia with reperfusion injury, we do 6 hours. We did pilot studies in the beginning trying to tease out what was the ideal time to have behavioral phenotype. If you do it too short, they don't care. And if you do more than 6 hours of occlusion, then it starts looking like the brachial artery occlusion. So we felt like 6 hours was kind of the sweet spot for this thing. Just tie the suture like if it was a shoelace, and at 6 hours, remove one stitch, pull the lace, and pull out the suture. It reperfuses really well.
And to overcome that problem, could you possibly electrically stimulate the muscle so that you increases the oxygen demand and make more ischemia?
If we would want to make the muscle, I could electrically stimulate it, but that would, I feel it would introduce a confounder, because you cannot cause muscle contraction without really getting to the nerves. So I would feel I would be stimulating also my sensory neurons. So, and then it really adds an extra layer that I think we would have to tease out better in order to do that.
You've been looking mostly at acute type bones, which you call necessarily chronic pain type of scale. A lot of the initial charts that you showed as far as behavioral analyses, mechanical thresholds and whatnot, it looked like they started to plateau, or some of them started to be declining. Is there a specific length of time using your techniques where they return somewhat to baseline levels?
So after 7 days, if you just do behavioral experiments, your animals look exactly the same as sham animals. It goes up to the resiliency of the mice, everything. I can tell you that gene expression does not come back. So that would be a different thing. I think what we're observing here is a little bit of this pain memory, if you want to put it like that. And that's why we observe the increased changes in behavior when we do our second injury. They are already primed for response. And if you're familiar with some of the experiments using hyperalgesic priming, where they give you a painful injury, and then they give a sub-threshold painful stimulus, but now your animals start responding, I believe it's more or less the same mechanism. You just have now more of these genes already available to respond when you get your second injury, and it just piles on top of that.
So, after recovery, a lot of times you get descending inhibition.
Mm-hmm.
Essentially, that masks the pain, and the animals can carry on about the business. Have you injected any intrathecal NQI or something to remove that mask to see if those pain responses return?
Nope. To be honest, we have never tried to dampen the descending inhibition. That's a great question, definitely. I believe that part of the recuperation time and how you see it, it definitely has to do with level of adaptation that you get on that side. But interestingly enough, muscle afferents are also very effective at rewiring your circuitry in the spinal cord and making it more sensitive to subsequent stimuli, and that's also believed to be one of the underlying causes on how chronic pain gets developed. So, okay, sure.
So your penetrating siRNA technique, you guys apply it a day or two before the injection.
Yes, we injected 2 days, at least for this siRNAs, we injected 2 days before the injury.
What happens, obviously, what happens if you apply that technique after the fact? I'm asking this in the context of if it could be used as a future treatment.
As a future treatment. Okay, so how interference, in this case, I believe it works, is just preventing the new protein being generated from the transcription. If you use it after the injury, all the new protein is already there and waiting, so I don't see it as a real tool that you could use as a potential therapeutic. It's not very nice, but I feel it's a very good tool to dissect the mechanisms, because you do not do a knockdown and just alter the whole system. You just bring it back to where it was before, so it's a little bit more functional in that sense.
But potentially you could use it maybe as a pre-surgical. Like, so we have a colleague, Dr. Nolan, who had both his knees replaced and the tourniquet pain. The knee replacement and all that function that worked fine. It was the tourniquet pain that was effectively replaced as far as the recovery. So if you could pre-inject some of those.
You're meeting it tomorrow, so looking forward to
That would be an interesting premise. I'm not sure how comfortable I feel about using gene manipulation therapy in patients. No, that's always a caveat. I mean, it's just a small interfering RNA, so it's on the lighter side of everything that you could do. I guess it could be teased out a little bit, because so far, I mean, we use it in animals, and we never think about what's the safety profile and the long term on those things. In children, we might have actually an answer for what's going on for presurgical pain, and we have done some studies looking at the role of growth hormone. You can, and it's interesting that I said in children's, because since we have found this very interesting growth hormone mediated mechanism of sensitization after surgical injuries, being growth hormone only works in children. It works when you actually need growth hormone in the system. Once you're an adult, it doesn't matter anymore. Just
Any last questions? We're at 2 o'clock, so
I have about 10 more questions.
you have your own special time. If you'd like to join me thanking Jose again.
Thank you very much, everyone.